Welcome to the webinar, Accelerating Drug Discovery for MASLD using High-Content Imaging in HEPATOPAC. Hi, I'm Etsuko Usuki, and I'll be facilitating the webinar today. I'm the Director of Services at BioIBT's lab in Kansas City, the legacy Xenotech site, where we offer a comprehensive portfolio of in vitro ADME research services and products. Before I introduce my colleague, Dr. Cottier, our speaker on today's webinar, I would like to take a few minutes to tell you about BioIBT. Founded in 1981, BioIBT has grown to become the largest provider of biospecimens and research services to the pharmaceutical and the diagnostics industry. We are dedicated to providing the services and the biospecimens that our customers need to help them advance their discovery, preclinical, and drug development programs. We have both grown organically and through strategic acquisitions that have enabled us to develop a comprehensive products portfolio and become the preferred provider for our global biopharmaceutical clients. As a legacy Xenotech scientist, I want to highlight that Xenotech joined BioIBT family in 2022, bringing its high reputation for ADME research services into BioIBT. The acquisition enabled us in the ADME services group to access BioIBT's extensive portfolio of biospecimens. BioIBT and Xenotech share a similar culture of scientific excellence and a passion for helping our clients achieve their research goals, which makes bringing our companies together a great fit. As a director at Kansas City site, I have over 25 years of experience working with our clients to design and implement drug metabolism studies that comply with the most up-to-date regulatory guidances and help them achieve their research objectives. One of our unique strengths is we partner with our clients to design studies using both conventional and custom methods, when conventional method may not be sufficient to answer our client questions. We can customize our studies to answer your specific research questions. For example, we can utilize the long-term HEPATOPAC model in our studies to evaluate low turnover and slowly transported compounds. The foundation of everything we do is a commitment to ethical sourcing, uncompromising quality, and a dedication to partnering with the research community. Helping our clients find solutions for their difficult questions is one of our favorite objectives. Here is a snapshot of the array of BioIBT's portfolio, including products used in cell and gene therapy programs, normal and disease state biospecimens, and the products and services to evaluate the ADME properties of drugs and prepare for IND submissions. Our ADME group has the industry's largest portfolio of hepatocytes, subcellular fractions, and recombinant enzymes, as well as other products to support ADME tox and DMPK research. Our team of experts works routinely with clients to help them identify hepatocyte lots, having the characteristics needed to meet their specific requirements. On the ADME services side, we work with our clients to design and conduct IND-enabling packages, including enzyme induction and inhibition, metabolism, drug transporter, as well as mechanistic studies to predict the pharmacokinetics of drug candidates. Our ADME services group provides individual studies as well as integrated programs based on our clients' need and challenges. Many of our clients start with consulting engagement, in which we provide expert advice to design ADME programs, assist with gap analysis, analyze existing data packages, or help prepare for regulatory meetings. We offer full in vitro ADME programs at our labs in Kansas City, Santa Clara, and Durham, North Carolina. When clients choose to work with us, they get the benefit of a team of experts who collectively have worked on thousands of programs and bring that experience to each new project. If you are interested in or have questions about the drug metabolism work that we do, I'll be happy to talk to you, but that will have to wait until another time. Additionally, we have partnerships that enable us to integrate in vivo studies into preclinical programs, and we have extensive capabilities for bioanalysis to support these programs or as standalone service. You can also visit bioibt.com to learn more about our portfolio of products and services. Now it is my pleasure to introduce Carissa Cottier. Dr. Cottier earned her PhD in medical pharmacology at the University of Arizona, where she focused on drug delivery in migraine headache model. In her postdoctoral fellowship at Johns Hopkins University, Carissa utilized molecular and histology-based assays in vitro model to study cerebral malaria. Following her postdoc, she joined BioIBT, where she leads our R&D programs in ADME products. Importantly, she led our program to develop high-purity Kupffer cells and develops new applications for our HEPATOPAC and HEPATimmune cultures. Carissa, I'm looking forward to your presentation.
Thank you, Atsuko, for that introduction. All right, so today I'm happy to be talking about a new application we've been developing in our HEPATOPAC model, looking at fatty liver disease kind of modeling, and especially highlighting the utility of high content imaging. So what is HEPATOPAC? HEPATOPAC is our long-term hepatocyte co-culture model. And so this is a pretty mature technology. It came out of MIT and was manufactured using microfabrication tools adapted from the semiconductor industry. And this allows us to create evenly spaced collagen islands. You can see that on the bottom left image. That is just a staining of those collagen islands. And then once we plate the primary hepatocytes onto those plates, they will preferentially stick to those islands, giving us the culture that you see in the top right. So this includes not only those primary hepatocytes from either human or preclinical species, but also a supporting stromal cell. And these supporting stromal cells are critical to the function of HEPATOPAC. It provides both the necessary cell-cell contacts as well as soluble factors to maintain the hepatocyte function long-term. One of the keys to this long-term function is the architecture, so the size and spacing between those hepatocyte islands. When this model was developed, they found that these factors were critical to the function. And so if you're looking in that middle graph, it's showing albumin and urea with different size and spacing formats. And we kind of settled on the kind of format shown in the gray bars that showed the highest function. So in addition to those general hepatocyte function markers, HEPATOPAC also maintains high drug metabolizing enzyme activity for up to 28 days. So you can see this is summarized in the table on the right, looking at HEPATOPAC at day 8 versus suspension cultures at day 0. And so we know that when hepatocytes are plated in a standard monoculture, the drug metabolizing activity quickly drops. However, in HEPATOPAC, this is maintained at a level similar to suspension hepatocytes. In addition to that drug metabolizing enzyme activity, HEPATOPAC is also maintaining that hepatic morphology that you would expect from healthy hepatocytes in the liver. So you can see that in both the human and preclinical species, is giving that nice polygonal distinct nuclei kind of format, as well as the notable biocanuliculi. And that is shown in the bottom left images, looking at the CD-FDA, as well as MRP2 protein staining. And that staining was done at 2 weeks into the HEPATOPAC culture. So still maintaining a very good morphology later in the culture. So just to close out kind of the intro in HEPATOPAC, today I'm going to be talking about kind of disease modeling specifically for fatty liver disease. But this model is actually developed more for looking at ADME-tox parameters, specifically to address low turnover compounds. And it has since expanded to kind of address many more research needs. And so if any of these other applications are of interest to you, we do have plenty of resources on our website for both HEPATOPAC and HepatImmune, which is just HEPATOPAC with those Kupffer cells. These resources are on our website. We have a number of training videos, posters, as well as our publication compendium. And so this compendium kind of compiles all of the peer-reviewed articles using HEPATOPAC and HepatImmune and separates them out based on application. So it's very easy to find the publications for the application that you are interested in. And so with that, I'm going to switch over to talking about fatty liver disease. And I wanted to give a very brief and simplified outline on fatty liver disease, just to give you an idea of how we ended up developing the media that we used for our studies. So you can kind of separate fatty liver disease into early stage disease and later stage disease. So early stage MAFLD is mostly just hepatic steatosis. It's very common. The last stats I've seen were suggesting that around 25% of the world population does have fatty liver disease. It's also often comorbid with other metabolic disorders and arises from a diet high in fat and sugar, especially fructose. And importantly, at this stage of disease, the kind of steatosis is reversible with lifestyle changes or management of some of those other underlying conditions. However, as this steatosis persists and progresses, the disease can kind of worsen into MASH. So this includes not only the fatty liver portion, but also inflammation and fibrosis. Eventually, if MASH persists in some patients, this can accelerate all the way to cirrhosis or even like liver carcinoma. So I've been giving this talk for a few years now, and it is exciting that I was able to update this slide. Previously, we did not have any approved therapeutics for MAFLD or MASH, but relatively recently, there was one therapeutic approved for MAFLD and MASH, and this is Rezdiffra, which is a partial agonist of THR beta. However, that does not mean that research has stopped for fatty liver disease. There are many other targets that are still currently under investigation, targeting diverse pathways, including de novo lipid synthesis, lipid metabolism, fibrosis, inflammation, any number of strategies. And to continue this research, one thing that is useful is to have in vitro models that you can use for kind of some of the drug screening and target identification and validation studies. And so these models have been around for a while. They often involve adding either exogenous free fatty acids, high glucose and fructose, or other factors, including cholesterol or inflammatory cytokines. In some cases, they do include some of those non-parenchymal cells, such as Kupffer cells and stellate cells. So it's kind of some of the options. They're all good options depending on your needs. A standard 2D culture, for instance, is able to load lipids quickly, certainly useful for some studies. You can incorporate those MPCs. However, as I mentioned, the longevity of those cultures is limited. So these are more suitable for shorter term studies. 3D microtissues such as spheroids are another good option. So they do have a longer term viability and function. Again, you can incorporate those MPC, and there are numerous studies published using these models. And finally, what I'm going to talk about today is HEPATOPAC and HEPATOMMUNE. Again, this does have that long-term viability and function. You can incorporate MPCs, but unlike the 3D spheroids, the handling is a lot simpler. Since it is kind of a flat culture model, you can handle it very similarly to 2D culture. And also because of this, it's a very simple to image. You do not require confocal imaging. So you can just kind of use your standard imaging setup to look at these cultures. And that is what we did for many of these studies. We used high content imaging to quantify lipid accumulation. So you can kind of see the hepatocyte island. This is just kind of a zoomed in view of one island with those supporting stromal cells. So if you look at the left, this is a culture which was incubated with free fatty acids. So bottom left image is just that bright field image. You can see those lipid droplets just using bright field. However, we wanted to be able to kind of better visualize and quantify these lipids. So we did use a neutral lipid dye, BOTA-B, which is shown in the next image over in green. The great thing about HCI analysis is that we can use the built-in analysis software to define that region of interest in the well. So that is the third image over from the left. And those white kind of areas are where we've trained the software to identify the lipids. And we did kind of set a threshold so that those hepatocytes with kind of their high background fluorescence were not counted in the analysis. And then finally, we generated a global or full well image so that we could quantify the lipid in the entire well. And then we could do this across the entire 96 well plate pretty rapidly. So this gave us quite a bit of data in a short amount of time. And so we did assess some different media formulations to induce that lipid accumulation. In the previous slide, we used free fatty acids. Here we did as well, along with high glucose and fructose, or a combination of both. Again, as I mentioned, these are some of the dietary factors that do lead to development of fatty liver disease. We did a time course for this, looking at days 2, 4, and 7, as well as kind of the reversal potential, where at day 4, we, in some cases, switched to a control media. And so we saw with the free fatty acid, as you might expect, this was a very quick lipid accumulation. That lipid staining is shown on the right graphs, as well as quantification of the triglycerides. And we saw this coming up right away. This is expected. There's not much signaling that needs to happen to kind of just load that lipid into the cell. Whereas when we looked at high glucose and fructose, it did take longer to kind of force that lipid accumulation. So we're seeing significant increases around day 7. And this is probably because that high glucose and fructose does need to go through de novo lipogenesis. And so the signaling might be a little bit more complex with that. We saw a kind of combination of that effect with combined free fatty acid and high glucose fructose, where we did see a high level of lipid right away that escalated by day 7. So in addition to the lipid accumulation, we wanted to assess some fatty liver associated gene changes to kind of see whether this model was recapitulating any of those seen clinically or in vivo. And so for these studies, we cultured in those 3 different medias for either 7 continuous days or 4 days, followed by a switch to control media at day 4 until day 7. So we collected those cell lysates, generated cDNA, and did a PCR array. And so just to note, most of the studies that I will be discussing today used our 96 well format. Excuse me. Or for these studies, we used our 24 well format to generate enough cDNA to perform this PCR array. So just to kind of look at some of the changes that we did see with those media, we looked at the FFA, HGF, and combination, both the downregulated and upregulated. And we saw kind of the most differentially regulated genes with the combination of FFA and HGF. We did feel that the HGF was providing perhaps a more complex model that is going a little bit more into the signaling regulation that's mimicking a fatty liver disease. And for our threshold, just for your information, we considered the genes significantly up or down regulated if they showed a 2-fold differential regulation from controls. And we did do 3 separate assessments in this array. And so I don't want to necessarily read out every gene that was differentially regulated with all of these media, but just want to kind of look at the different media and compare to either published clinical data or in vivo data to kind of see where we are in the disease progression for these different medias. And as mentioned, don't worry if you do want to take a closer look at this. We will provide these slides and the recording after the session. But just kind of looking at genes that were upregulated, for instance, with free fatty acid treatment. If you look at the list, for instance, with CD36, we are looking at kind of an earlier stage disease where these changes are recapitulated in MAFLD patients or just with a high-fat diet in mouse, for instance. So we're at that earlier stage, maybe with MAFLD not progressing toward MASH for the free fatty acid treatment. Again, that's true as well with the genes that were downregulated, just showing an example of an in vivo data, for instance, at the bottom right, which recapitulates that, yes, some of these genes are downregulated with a high-fat diet in rats. So if we look at genes that were differentially regulated with that high glucose fructose treatment, we do see some indication that these genes are similarly regulated and kind of regulated at more advanced stages of fatty liver disease. So those graphs on the right are from a peer-reviewed publication looking at both at the top a rat model feeding with high fat diet or fructose, and at the bottom looking at clinical samples. So 2 of these genes that we saw differentially regulated were also upregulated in NASH, but not just hepatic steatosis. And again, similarly with downregulated genes, I do want to point out that unlike in clinical samples, where they see upregulation of CYP2A1, we did not. We saw downregulation. So this is kind of matching more closely with studies that were using high fructose in mouse. So we're not quite hitting that clinical change, but we are recapitulating in vivo. And finally, looking at free fatty acid and high glucose fructose treatment, we did see the largest variety of genes that were upregulated. And again, looking at some of these genes, we do see examples where they are upregulated, more associated with later stage disease, such as MASH, and kind of correlating to clinical MAFLD samples. And closing out again with downregulated genes, we did once again see downregulation of CYP2A1. And then the rest of these genes were kind of correlated with either rodent models or clinical models. Just to point out, IGF-1, for instance, showed a lower expression in clinical samples with a higher NAS score. And that NAS score is just the clinical kind of histopathological method of ranking fatty liver disease. Okay, so after we established the model, kind of the basic parameters of can it load lipids and are there the expected kind of gene changes, we did want to test this model for its ability to be used in screening. So our first step was just screening small molecule drugs for their lipid lowering abilities. In these studies, we used 3 different ACC1 and 2 inhibitors, and at first did a study where we pre-treated the cultures with these compounds and then added the free fatty acid to see if they could prevent the steatosis. And we do see that they were able to prevent the steatosis in a concentration-dependent manner. However, a more realistic dosing schedule would be to actually start dosing these compounds after disease has already been established. So for these studies, we loaded the cultures with lipids for 4 days ahead of time and then treated with these compounds while we still continued to treat with that free fatty acid. And we were also able to see the ability of this model to show reversal of that steatosis, again, in a concentration-dependent manner. So switching gears a little bit, there's also the potential for the opposite problem, where drugs can actually induce steatosis. And this is a very specific mechanism of drug-induced liver injury called drug-induced steatosis. So we assessed valproic acid. This is a compound that is known in some cases to cause hepatic steatosis. We assessed over a number of different concentrations, with our lowest concentration falling just below the clinical Cmax for valproic acid. And so in lean cultures, just using the control media and valproic acid, we didn't really see significant lipid accumulation until we got to the very highest concentration. And that is a very high concentration and not very clinically relevant. However, if we had established disease with either free fatty acid or high glucose fructose ahead of time, and then continued to also treat with valproic acid, we saw essentially a worsening of disease at that very lowest concentration. Again, that does fall below the clinical Cmax for valproic acid. This kind of shows that you may see differences in drug-induced steatosis and at what concentration you are seeing that, depending if you're using a lean culture or kind of a disease-relevant culture. And importantly, we actually did not see any difference in drug toxicity as far as ATP. It was only related to that steatosis accumulation. So next I want to switch gears and talk about genetic variants in MAFLD. So we know that there are a number of different SNPs that have been shown to either increase fatty liver disease risk or reduce it. So we do have hepatocytes that are genotyped for some of these risk and kind of rescue alleles. So we are currently genotyping our hepatocytes for the PNPLA3 mutation, as well as the HSD17 beta 13 mutation. And so the next few studies I will be talking about will be using hepatocytes that were genotyped for the PNPLA3 mutation, and that does increase the risk for fatty liver disease. So we had hepatocytes from donors that express the mutant PNPLA3, that's a I148M SNP, as well as wild type. And we did find that those donors in HEPATOPAC are more sensitive to free fatty acid than the wild type. So we used 3 different donors for each genotype and looked at escalating concentrations of free fatty acid. Again, we did both loading as well as unloading, where we switched to control media. And so we saw both that the hepatocytes with that PNPLA3 mutation had increased lipid accumulation, as well as a reduced ability to kind of clear that lipid after the free fatty acid had been removed. And so the mechanism for this is kind of interesting. So it's thought that that mutant PNPLA3 protein is accumulating on the lipid droplets and preventing their lipolysis. So it's not exactly a loss of function, it's more blocking the lipolysis. So we wanted to know what would happen if you knock down PNPLA3 in those genotyped hepatocytes. So to kind of accomplish this, we did need to figure out a mechanism for doing siRNA knockdown in HEPATOPAC. If you did attend our webinar 2 weeks ago, you saw more in-depth kind of explanation of this method. And if you are interested, do feel free to go back and watch that webinar. But essentially, we are able to knock down genes just specifically in the hepatocytes by doing this knockdown at D0. So just a little info about the manufacture of HEPATOPAC. At D0, we're seeding those primary hepatocytes, and it is not until day 1 that we're adding in those supporting stromal cells. So we do have about a 24-hour period where we can expose the hepatocytes specifically to an siRNA, and that is what we've done. So for this proof of concept, we used CYP3/4, and we were able to look at both the protein activity, looking at midazolam formation of a metabolite, as well as mRNA knockdown. And we were able to see by both methods about an 80% knockdown that was sustained until at least day 18 of culture. So when we knocked down PNPLA3 using siRNA, we saw something kind of interesting. We saw that when we were treating with FFA, the siRNA knockdown was reducing lipid accumulation only in those mutant donors. So you can see in the graph on the right, the quantification of the lipid accumulation. In the wild type donors, the knockdown made no difference. However, in the mutant donors, we did see a significant reduction in lipid accumulation. And this does kind of make sense if you think back to the proposed mechanism where that PNPLA3 protein, when it has that I148M switch, is kind of tethered to those lipid droplets and preventing their lipolysis. That is not the case in the wild type protein. So once you're getting rid of that block on the lipid droplets, you're able to, again, induce lipolysis of these lipids. We also wanted to look at a genetic variant that reduced fatty liver disease, and we were interested in CIDEB, specifically because we wanted to do some additional analysis looking at reduced lipid droplet size, as has been reported. We couldn't really genotype donors for these particular mutations. It is loss of function mutations that are reducing fatty liver disease. However, there are many, many different SNPs associated, so it was difficult to kind of hone in on which ones would be important. And for this, we again used high content imaging. We were able to do some more in-depth analyses to measure lipid droplet size. This was kind of fun. We still used that PhotoB stain, which is a neutral lipid stain, but had to apply some filters to really get, crisp lipid droplets, and then define that as a region of interest for calculation. And so we were able for this to count each droplet as a unique quantification of the object radius as well as area. And as expected, when we knocked down CIDEB, we both reduced lipid accumulation as well as droplet size. So for these studies, we used both free fatty acid as well as high glucose fructose. And for both of these methods, we saw similar results. We did see a good level of knockdown of CIDEB from start to finish for this study. You can see in that first graph on the left, we looked at mean droplet area, and this was reduced with the knockdown. Even in the non-treated controls, actually, there is a little bit of lipid that will start to accumulate over time in these cultures. And for those, even the lipid droplets were smaller. We looked at lipid accumulation in that middle graph. We saw significantly reduced lipids when we did the knockdown. And then kind of the sum of area per island area as well, we saw reduced lipid accumulation. So what we're seeing is that when we do this knockdown, we're preventing the formation of those very large lipid droplets. So for this analysis, we looked at lipid droplets in a specific field, measured them, and placed them into size range bins. And this is relative frequency, so it is normalized, as the CIDEB knockdown did also reduce the number of lipid droplets. So that was important to kind of fairly assess this. So the CIDEB siRNA is shown in red, and the skirmal control is shown in blue. And I think you can appreciate that those lipid droplets are far shifted to the left with that knockdown. And just to close out, I want to touch on some of the preliminary work we've done using the HEPATOPAC platform. So up until now, I've been talking just about the HEPATOPAC platform. Again, those supporting stromal cells, as well as the primary hepatocytes. But we do have the ability to add Kupffer cells as well. So initially, this model was kind of developed to look at immune-mediated drug toxicity. And so the advantage of adding Kupffer cells, these are the resident macrophages of the liver, and they're going to be the first cells that can encounter invading particles and drugs into the liver, and their activation does lead to release of inflammatory mediators. This can also be useful for our fatty liver disease model. We've done some validation of this model, looking at Kupffer cell number, just staining for Kupffer cell markers and macrophage markers, as well as their function over time. when we're looking at a long-term model, it is important that these cells are able to remain constantly functional and able to respond to insults over a longer period of time. And so we did see, again, we used high content imaging to count these cells and identify them based on CD68 staining. We found that they were replicating very slowly. So over the course of the culture, maybe by day 13, they doubled. So it's pretty slow. We also looked at response to LPS and measured IL-6. That's on the right graph, looking at HEPATOPAC in teal and hepatimune in the green. Do see a lot more response when we're adding those Kupffer cells. And this ability to respond to LPS was maintained for 13 days after the Kupffer cells were added. We didn't assess days after that, so they could be functional for even longer. So other people have also looked more in depth at a variety of cytokines released from LPS-stimulated Kupffer cells, specifically in hepatimune, and looking at different Kupffer cell ratios to kind of see the cell concentration response. So looking at IL-1 beta, IL-6, IL-8, 10, TNF-alpha, and interferon gamma. And so in all cases, these cells were responding as expected. And as you increase the cell number, you increase the response. And so what we're going for when we're adding cells is kind of mimicking, I guess, a diseased liver where the amount of Kupffer cells is going to be increased. So moving back into fatty liver disease, we do know that inflammatory activation of Kupffer cells has been implicated in MAFLD and MASH in vivo. And so, for instance, it's been shown that LPS injection in vivo will increase hepatic steatosis, and depletion of Kupffer cells in vivo will protect against high-fat diet or sucrose diet-induced liver steatosis. It's been shown that a high-fat diet will induce Kupffer cells to release inflammatory mediators such as TNF-alpha, and this will lead to increased free fatty acid loading in cultured hepatocytes. We also know from publications that fatty acids can regulate the Kupffer cell M1, M2 polarization. So this is looking at Kupffer cells responding to different dietary saturated free fatty acids. So this will produce inflammation. So in these studies, palmitic acid was used. This is also used as part of our FFA formulation. And this is a dietary saturated fatty acid. This is what you think of when you're thinking of like bad fats. And so they found that this is increasing inflammatory expression and M1 markers in Kupffer cells. Whereas when they use DHA, which is an N3 polyunsaturated fatty acid, again, this is more of what you hear in the kind of just general media as a good fat. This had the opposite effect. It was anti-inflammatory. So we did do just one study looking at lipid accumulation and reversal, again, with and without Kupffer cells. So for these cultures, we were just assessing one time point. That's day 7. So on the left, we're looking at lipid accumulation at day 7 in HEPATOPAC or hepatimune. And then those middle panels, we're looking at, again, lipid unloading or switch to control to see the reversal potential. And so we did see for both FFA and HGF an increase in lipid accumulation in hepatimune compared to a HEPATOPAC. This appeared to be true with the FFA plus HGF as well. And then interestingly, in addition to that, we did see in that middle panel that the hepatimune was more resistant to reversal once we switch back to that lean control media. And these are quantified in the graphs to the right. Just to close things out, we We've done some studies to show that HEPATOPAC and HEPATIMMUNE can be used for disease modeling, drug toxicity studies, focused on a variety of aspects of fatty liver disease. One of our focuses has been using siRNA to examine the influence of gene knockdown on lipid limiting parameters. And that can be used, for instance, for any of your drug compounds that are siRNA-based. We can use those as well. We do not just have to use commercially available siRNA. So as I mentioned, we can not only assess your siRNA drug candidates in our standard HEPATOPAC, but also look at the effects in different genotypes using donors that have some of those risk or rescue phenotypes. We did just one study showing that the HEPATIMMUNE model can add more of an immune cell component and increase that fatty liver assessments and lipid accumulation. Okay, with that, I would be very happy to take any questions. If you do end up wanting to get in touch with me, do feel free. My contact info is below. And thank you for your attention.
Thank you very much, Carissa, for a very interesting presentation today. We have now entered the Q&A session. If you haven't already, please post your questions in the questions pane. Please note that we have an array of HEPATOPAC resources available on our website. Visit the resource page and search for HEPATOPAC and education content to find the training videos, user guides, brochures, and publications. Let's see. We have a few questions in. The first one, can HEPATOPAC be used with GalNAc conjugated siRNA for delivery by free uptake instead of transfection?
Yeah, thanks for that question. I didn't talk about it here, but we have done some studies, not necessarily related to fatty liver disease, but just in general using GalNAc siRNA. It is very amenable to those studies. We've done time courses looking at ASGPR expression and siRNA uptake. And we are actually now validating our hepatocyte lots for their ability to be used for GalNAc. So we're characterizing all of our new platable lots for their ability to knock down a target of interest using a GalNAc siRNA. So yes, absolutely, this model can be used for that purpose.
Okay, great, thank you. Another question, can we collect samples for other endpoints from the sample wells when doing a high content imaging for lipids?
Yeah, so the focus of the talk today was high content imaging, but it is amenable to other endpoints. The easiest thing that you could do with the high content imaging is collection of supernames. So looking at any soluble factors, inflammatory mediators, hepatocyte function, or anything else that you're interested in. When we are doing this staining, we tend not to collect like lysates from the same wells that were stained, but you can, when you're doing that lipid staining, kind of stain for other markers. It is amenable, for instance, to co-staining with antibodies for other specific targets.
Interesting, thank you. I think we have a bit more time for a few more questions. If I have a hepatocyte lot that I'm using for other studies, can I use it in HEPATOPAC?
Yeah. So we do have lots that we've used often in HEPATOPAC and kind of validated for drug metabolizing enzyme activity or just like baseline lipid accumulation. But pretty much any platable lot is able to be used in HEPATOPAC, so that certainly would not be a problem. Absolutely.
Okay. Good. think that's going to be the last question. If there's nothing else, do you need to pull samples from multiple wells to look at the gene expression?
Yeah, so I kind of touched on that when I talked about the array. So for just general PCR of like single targets, we used 96 well plates, and we didn't actually have to pool our samples. We have sufficient RNA. For the array, to get around that issue, again, we did use the 24 well format just to get more cells and more RNA. But I guess if you're looking at a low abundance mRNA, you might have to, and if you're doing something more like bigger, like RNA-seq, again, you might have to pool samples just to get sufficient material. But if you, do want to do those applications, definitely reach out. We have protocols for kind of preparing samples for those.
Great. Thank you very much. I think that was the last question we want to just go over right now. But well, I guess we can conclude the Q&A session. But as a reminder, this is the fourth in a series of 5 webinars. If you missed any of the previous ones, they are now available on demand on your website, on our website. If you haven't already, please register for the next webinar, the last one on November 6th, in which Jean-Marie Gaffney will discuss conducting reaction phenotyping studies for low turnover compound using a HEPATOPAC. I think this is going to be a very interesting topic.
Yes, absolutely.
Thank you very much. Thank you, Carissa, and thank you very much for your attendance today. As mentioned earlier, here's my email address as well as Carissa's. You are welcome to contact us directly. You can also contact us via the website at the address shown. You will be receiving a short survey to provide you feedback on this webinar and how we can better serve you in the future. Thank you again for your attendance.
Yes, thank you. Have a great day.